For most of human history, entire populations lived and disappeared without leaving names, cities, or written records. What we know about them comes from fragments that survived by chance. In this video, we focus on one of those populations. They were not identified through monuments or stories, but through DNA found in a small cave in Siberia. Over time, that discovery changed how scientists understand human history in Asia. The Denisovans were real people, distinct from Neanderthals and modern humans, yet closely connected to both. Their traces appear in ancient bones, in soil, and even inside the genomes of people alive today. This documentary does not tell a legend or a lost myth. It follows the evidence that exists, and it stays within what the record allows. What remains is a quiet reconstruction of a population known only through traces that time did not fully erase. Quiet evidence in a cold cave. In the Altai Mountains of southern Siberia, a limestone chamber sits above the Anui River. The place is known as Denisova Cave, after a local hermit tradition attached to the site. Archaeologists began systematic work there in the late 20th century, using careful excavation squares, fine sieves, and layer-by-layer recording. The cave holds long sequences of sediment, including fragments of stone tools, animal bone, charcoal, and occasional human remains. Many pieces are small and unremarkable on first sight. Their value comes from the order in which they appear, and the dates that can be tied to each layer. In 2008, excavators recovered a tiny bone from a layer associated with the Middle to Upper Paleolithic transition. The piece was a distal phalanx, a finger bone, from a juvenile. It was stored and catalogued with other finds, then later selected for genetic work. The specimen carried no clear anatomical marker that could separate it from an early modern human or a Neanderthal. Its importance arrived through laboratory methods rather than through its shape. In 2010, a research team led by Svante Pääbo at the Max Planck Institute for Evolutionary Anthropology reported a genome sequence from the bone that did not match the genetic profile of Neanderthals or present-day humans. The paper did not introduce a named civilization or a mapped population center. It described a distinct group of archaic humans, identified through DNA, and it did so with narrow language tied to testable comparisons. The group later became known as Denisovans, a name drawn from the cave rather than from a known historical self-designation. The earliest public descriptions stayed close to laboratory facts: base pair differences, contamination controls, and statistical relationships to other genomes. The calmness of those initial reports can be felt in their emphasis on what could be measured, repeated, and compared across independent samples. Denisova Cave then produced additional material. A large molar tooth, found in earlier seasons, was also sampled. Its genetic signal aligned with the new group. Other layers contained small bone fragments that could not be assigned by morphology alone, and some were later tested. Over time, the cave became less a single discovery site, and more a reference point for a new category of human variation. The work broadened into sediments as well. In some later studies, DNA fragments extracted directly from cave sediments revealed the presence of Denisovan genetic material, even when no identifiable bones were present. This approach changed the practical meaning of absence. An empty square, if it preserved DNA in the soil, could still carry information about who had been there. The geographical story did not remain confined to Siberia. As more modern human genomes were sequenced, researchers began to detect Denisovan ancestry in living populations. The highest proportions were reported in parts of Oceania, including Papua New Guinea and nearby island regions. Smaller signals appeared in parts of East and South Asia. These findings did not require a claim of a lost city or an engineered system. They rested on inherited segments of DNA that could be modelled as the result of ancient interbreeding between Denisovan groups and anatomically modern humans. A separate line of research involved high-altitude adaptation. On the Tibetan Plateau, certain genetic variants help with life in low-oxygen environments. A well-studied example is a variant linked to the EPAS-1 region, associated with reduced hemoglobin concentration in high-altitude populations. Genetic analyses have indicated that this variant likely entered the modern human gene pool through admixture with a Denisovan-related group. The statement remains technical and limited. It points to gene flow rather than to institutions, governance, or a constructed landscape. Evidence for Denisovan presence outside Siberia also came from the Tibetan region itself. In the 20th century, a large human mandible was found near Shahe, in Gansu province, close to Baishia Karst cave. The specimen's age was evaluated using methods suited to its context, and for a long time it was discussed without a clear taxonomic label. In 2019, researchers used protein analysis from dental calculus and bone to associate the mandible with Denisovans. The result did not expand into a full narrative. It provided a location, a specimen, and a biochemical link to the genetic group first defined at Denisova cave. The archaeological record in Denisova cave includes artifacts that are sometimes described in simple terms as refined or unusual for their age. A polished stone bracelet, made of green chloritolite, found in a layer dated to around 40,000 years ago, has been discussed in reports and museum displays. The object shows a drilled hole and a finished surface. The cave also yielded bone points and ornaments, including beads. Attribution remains careful when written by specialists. Objects are associated with layers, and layers may contain traces of multiple groups over time. Neanderthal remains, modern human remains, and Denisovan genetic signals have all been identified in different strata, and some layers show evidence of overlap or close succession. One of the clearest factual features of Denisova cave is that it preserves a long and complex occupation history. Neanderthal DNA has been recovered from the site, and fossils assigned to Neanderthals have been found there. A notable individual, identified through genetic testing, was a first-generation offspring of a Neanderthal mother and a Denisovan father. The specimen is often referenced as Deni, based on its laboratory label, and it illustrates that contact between groups was not rare in purely statistical terms. This fact does not lead by itself to a broader account of social structure. It simply shows that interbreeding occurred, and that the genomes can record it. Time depth is another defining feature. Denisovan-related signals are present in samples that suggest a wide range of dates, from hundreds of thousands of years ago to the period when modern humans were expanding across Eurasia. Dating in caves is usually done through a combination of methods, including radiocarbon dating for younger layers, optically stimulated luminescence for sediments, and uranium series methods for certain mineral contexts. Each method carries its own limits, and careful papers tend to report uncertainties rather than fixed points. The result is a timeline that remains bounded by error margins and by the availability of material that can be dated. The term Denisovan can sound like a complete identity, but it is a label created to describe a genetic cluster found through modern techniques. It does not point to a known language, a recorded name, or a single region of origin. The present evidence suggests a broad and varied presence across Asia, inferred through DNA and through a small number of fossils. Researchers often speak of Denisovan-related populations rather than a single group, because the genetic differences among Denisovan sequences imply diversity across time and space. That diversity can be modelled, but it cannot yet be mapped in full detail using only the few physical remains currently available. Some modern accounts attach large claims to this limited record. The facts in the published literature are smaller and more specific. A finger bone in Siberia, dated through its sediment context and studied in a Leipzig laboratory, provided a first genetic signal. A mandible on the edge of the Tibetan plateau added a second anchor point, this time through proteins. Patterns in present-day genomes extended the map indirectly, showing where Denisovan ancestry persisted. These elements stand without needing a complete story built around them. They form a set of constraints. Denisovans existed. They were distinct from Neanderthals and from modern humans. They interacted with modern humans, and they occupied parts of Asia across long periods. The archaeological ground remains quiet on its own terms. Sediment accumulates, animals die, stones chip and scatter. In the cave, the smallest pieces may carry the most information, not because they tell a full history, but because they can be tested. That is the condition of Denisovan research at present. The record is real, the samples are few, and the picture is assembled through methods that favour careful edges over large declarations. Names written. In DNA, not stone. The study of Denisovans moved forward without the discovery of villages, burial grounds, or carved markers. Instead, it advanced through comparisons between genomes. After the first Denisovan genome was published in 2010, researchers began using it as a reference point. They compared its sequence to those of present-day humans from different regions. The method relied on identifying short segments of DNA that were more similar to the Denisovan genome than to sequences from African populations, which served as a baseline for modern human variation before admixture events outside Africa. Early results showed a pattern that was unevenly distributed. Populations in Papua New Guinea, Bougainville, and parts of the Bismarck archipelago carried the highest proportion of Denisovan ancestry, sometimes estimated at several percent. East Asian populations showed smaller but still detectable traces. South Asian populations displayed faint signals in some datasets, while most European populations showed little to none. These differences were not presented as cultural markers. They were treated as inherited biological fragments, passed down through reproduction and shaped by later population movements. The identification of Denisovan ancestry required statistical tools designed to separate ancient admixture from more recent shared ancestry. One commonly used approach involved D-statistics, sometimes referred to as the Abba-Baba test. In practice, the analysis did not identify individuals as Denisovan descendants. It measured probabilities across populations, producing values that suggested ancient interbreeding rather than direct lineage continuity. As datasets grew larger, researchers refined their models. Whole genome sequencing allowed for the detection of specific introgress segments rather than broad averages. These segments varied in length, with shorter fragments generally indicating older admixture events due to recombination over time. Longer segments suggested more recent contact. In Denisovan-related analyses, many detected segments were short, consistent with interactions tens of thousands of years in the past. This temporal distance limited the precision of reconstructions, but strengthened the case that the admixture was ancient rather than recent. Some Denisovan-derived segments were not randomly distributed across the genome. The EPAS1-related variant in Tibetan groups became a frequently cited example. Studies showed that this variant matched a Denisovan sequence more closely than any known modern human version. Functional studies linked the variant to physiological responses to low oxygen, but genetic papers maintained a restrained tone. The finding was described as adaptive introgression, a process by which a gene from one population confers a survival advantage in another after interbreeding. Outside of high-altitude adaptation, most Denisovan-derived segments did not have clear functional interpretations. Their presence was documented without assigning purpose. Researchers emphasized that introgression does not imply advancement or decline. It reflects contact between groups and the random survival of certain genetic segments across generations. The geographical pattern raised questions that were framed cautiously. Denisova Cave is located in southern Siberia, far from Oceania. Yet the highest Denisovan ancestry appeared thousands of kilometers away. To address this, researchers proposed that Denisovan-related populations were once widespread across Asia. The fossil record to support this remained sparse, but the genetic evidence suggested multiple Denisovan groups rather than a single uniform population. Some studies proposed at least two distinct Denisovan lineages, inferred from differences between Denisovan segments found in East Asian populations and those found in Oceanian populations. These proposals were built from comparative models rather than direct observation. They did not rely on artifacts or settlement patterns. Instead, they used differences in sequence divergence and segment distribution to infer population structure. The language in these studies remained conditional. Words like likely, consistent with, and suggests were used to mark the boundary between data and inference. No formal naming system was applied to the proposed Denisovan subgroups, reflecting the absence of physical reference points. The Denisovan genome itself also showed complexity. It carried evidence of admixture with Neanderthals, indicating that Denisovans were not isolated from other archaic humans. In some analyses, Denisovans also appeared to carry genetic material from an even older hominin population, sometimes described as a ghost lineage. This conclusion came from patterns of deep divergence that could not be explained by known Neanderthal or modern human ancestry. The proposed population was not identified through fossils. It was inferred mathematically, Through mismatches in expected genetic distances, such findings expanded the picture without solidifying it. The genetic landscape of ancient Eurasia appeared layered, with multiple groups overlapping in time and space. Denisovans occupied a position within this landscape that was defined by relationships rather than by physical boundaries. Their identity existed as a set of comparisons, closer to Neanderthals than to modern humans, yet distinct from both. The technical nature of this work influenced how results were presented. Papers focused on sampling methods, sequencing, depth, error rates, and contamination controls. Charts displayed allele frequencies and confidence intervals rather than maps of territory. When geographical terms were used, they referred to the location of sampled modern populations, not to reconstructed Denisovan homelands. This approach limited the scope of interpretation and kept the emphasis on what the data could directly support. One consequence of relying on modern genomes is that the record is filtered by survival. Denisovan ancestry is only visible where it persisted through many generations of population change. Regions where Denisovan-related populations once lived but left no genetic trace in modern people remain invisible to this method. Researchers acknowledge this limitation in method sections and supplementary materials, noting that absence of evidence in genomes does not equal evidence of absence in the past. The name Denisovan continued to function as a technical shorthand. It referred to a genome assembled from fragments found in a Siberian cave and extended through statistical resemblance elsewhere. No inscriptions, oral traditions, or symbolic systems could be associated with it. The identity was written in base pairs, not in stone or pigment. Each new dataset refined the outline slightly, adding resolution in some areas while leaving others blank. By the mid-2010s, Denisovans were no longer a singular anomaly. They became a standard reference in discussions of human evolution in Asia. Their presence was assumed in models of migration and admixture, even when the details were left open. The field adjusted to the idea that the human past included multiple interacting populations whose traces survived unevenly. The work remained ongoing, incremental, and constrained by available material. New genomes added data points rather than sweeping changes. The Denisovan name stayed attached to a method of detection rather than to a reconstructed society. What persisted was a pattern. Fragments of ancient DNA, measured and compared, pointing to contact, mixture, and disappearance without requiring a narrative beyond the evidence itself. Tools without names, and layers without voices. The archaeological record connected to Denisovan research does not begin with a defined toolkit or a recognized cultural package. In Denisova Cave, excavators divided the deposits into finely measured units, recording depth, composition, and association with nearby materials. Each layer reflects a period of accumulation shaped by natural processes and intermittent human presence. Stone fragments, animal bones, and traces of fire appear unevenly across the sequence. The record does not form a continuous narrative. It consists of intervals separated by erosion, collapse, and long spans of minimal activity. Stone tools recovered from Denisova Cave span a wide time range. Some belong to Middle Paleolithic industries, typically associated elsewhere with Neanderthals. These include simple flake tools, scrapers, and cores produced through prepared techniques. Other layers contain artifacts often described as Upper Paleolithic in character, such as blades, bladelets, and worked bone. Archaeologists treat these categories as descriptive rather than definitive. The presence of a tool type does not assign authorship to a specific group unless supported by additional evidence. This caution is necessary because Denisova Cave preserves remains from more than one human population. Neanderthal fossils and DNA have been recovered from certain layers. Modern human remains have also been identified in others. Denisovan genetic material appears in multiple parts of the stratigraphic sequence, sometimes overlapping in time with the others. As a result, tools found in Denisovan-associated layers are not automatically labeled as Denisovan tools. They are described in relation to their form, material, and manufacturing technique, not to a named group. Among the most frequently discussed objects from the cave is a polished stone bracelet fragment made of green chloritolite. The piece was recovered from a layer dated to roughly 40,000 years ago, using a combination of radiocarbon dating on associated materials and stratigraphic correlation. The bracelet shows a drilled hole with smooth edges and a finished surface. Microscopic analysis suggests polishing with fine abrasives. Reports describe the object in measured terms, noting its material properties and manufacturing traces without assigning intent or symbolic meaning. Bone artifacts from the site include points and fragments shaped through grinding and scraping. Some are interpreted as tools, others as ornaments. Perforated animal teeth and beads made from bone and shell have also been found. These items appear sporadically rather than as part of a dense assemblage. Their distribution suggests episodic use of the cave rather than long-term occupation. The materials used often came from local sources, consistent with mobile populations moving through the region rather than maintaining permanent settlements. Dating these artifacts relies on the surrounding sediment rather than on the objects themselves. Organic materials suitable for radiocarbon dating are rare in deeper layers. Where possible, charcoal fragments are dated directly. In other cases, optically stimulated luminescence dating is applied to sediments to estimate when mineral grains were last exposed to light. Each method introduces uncertainty, which is reported as a range rather than a single year. Archaeologists work within these ranges, aligning tool types with approximate periods rather than fixed timelines. Beyond Denisova cave, the material record associated with Denisovan-related populations remains sparse. The Shahe mandible, linked to Denisovans through protein analysis, was not found with a clear tool assemblage. Its context suggests occupation of the northeastern edge of the Tibetan Plateau, but the surrounding archaeological record does not provide a distinct technological signature. Stone tools from the region span broad periods and are difficult to assign to specific populations without genetic material. In Southeast Asia and Oceania, where modern populations show high levels of Denisovan ancestry, direct Denisovan fossils have not been identified. Archaeological sites in these regions contain stone tools and other materials attributed to early modern humans. Genetic evidence suggests interaction between Denisovan-related populations and modern humans, but the material record does not clearly separate their contributions. Tools are described according to established regional traditions, and Denisovan presence remains inferred rather than directly observed. One reason for this limitation lies in preservation. Tropical environments tend to degrade bone and DNA more rapidly than colder regions. As a result, fossil evidence is less likely to survive long enough to be recovered and analysed. Stone tools endure, but they carry limited information about the biological identity of their makers. Without associated human remains or genetic traces in sediment, attribution remains uncertain. Archaeologists therefore focus on describing assemblages without naming their authors. Reports emphasize reduction strategies, raw material selection, and wear patterns. These features can be compared across sites to identify similarities and differences, but they do not resolve questions of population identity on their own. A blade produced by a Denisovan-related individual would be indistinguishable from one produced by a modern human using the same technique. The Denisovan case has encouraged the use of sediment DNA analysis at archaeological sites. By extracting genetic material directly from soil samples, researchers can sometimes identify which human groups were present even when bones are absent. In Denisova cave, this method revealed Denisovan and Neanderthal DNA in layers that contained no diagnostic fossils. The approach remains technically demanding and is sensitive to contamination and preservation conditions. Results are reported cautiously, often with multiple controls to confirm authenticity. Sediment DNA does not provide information about tools or behavior directly. It establishes presence, not activity. A layer may contain Denisovan DNA and stone tools, but their association still requires interpretation. Archaeologists avoid simple equations, recognizing that materials can be moved by natural processes or reused by later occupants. The cave environment itself complicates matters, as sediments can be disturbed by water flow, animal burrowing, or human activity. The broader archaeological picture that emerges is one of incomplete alignment. Genetic data identify Denisovans as a distinct population. Archaeological data show complex tool traditions across Eurasia during the relevant periods. The two records intersect only in specific contexts, such as Denisova cave, where both types of evidence coexist. Even there, the association remains layered rather than unified. Researchers continue to publish site reports that expand the dataset incrementally. Each layer is documented with depth, composition, and associated finds. The absence of a defined Denisovan material culture is treated as a factual condition rather than a problem to be solved. It reflects the limits of current evidence and the nature of Paleolithic archaeology. The tools remain, but they do not carry names. The layers preserve sequences, not voices. What can be said is restricted to what is preserved, measured, and compared. Denisovan-related archaeology exists within this framework, as a set of contexts where genetics and material traces occasionally overlap, without forming a complete or unified cultural record. A presence spread thin across Asia. The Denisovan record does not point to a single center or a compact homeland. Instead, it appears as a dispersed signal, detected unevenly across a wide portion of Asia. This pattern is not drawn from settlement maps or site networks. It is reconstructed from genetic traces, a small number of fossil finds, and the environmental contexts in which those traces appear. Each line of evidence is partial. Together, they outline a presence that was broad but lightly recorded. The first physical reference point remains Denisova Cave in the Altai Mountains. The region sits at a junction of ecological zones where steppe, forest, and mountain environments meet. During the Middle and Late Pleistocene, climate shifts repeatedly altered local conditions. Cold phases expanded grasslands, while warmer periods allowed forests to advance. Such variability supports intermittent human use, rather than permanent occupation. The cave's long sediment sequence reflects this pattern, with gaps and returns rather than continuous habitation. From this northern location, the Denisovan signal extends south and east through genetic inference. Modern populations in East Asia show low but consistent levels of Denisovan-related ancestry. These signals are weaker than those found in Oceania, but remain statistically significant in large datasets. Their distribution suggests that Denisovan-related groups were present somewhere along the migration routes of early modern humans moving eastward. The exact locations of contact remain unspecified, as genetic data alone cannot identify meeting points on the landscape. The strongest genetic signals appear in Melanesia and parts of island Southeast Asia. Populations in Papua New Guinea and nearby islands carry the highest known proportions of Denisovan ancestry. This finding initially appeared difficult to reconcile with the limited fossil record, which offers no Denisovan remains from these regions. Researchers addressed the discrepancy by proposing a wide Denisovan range across mainland Asia and possibly into parts of Southeast Asia before the arrival of modern humans in those areas. The proposal rests on demographic modelling rather than direct observation. Environmental context plays a role in these reconstructions. During periods of lower sea level, land bridges connected parts of Southeast Asia that are now separated by water. These connections allowed human populations to move across regions that later became islands. Denisovan-related groups, if present in these landscapes, would not require advanced seafaring to reach areas that now appear remote. The archaeological record in these regions, however, does not preserve clear markers of Denisovan activity, leaving the reconstruction dependent on genetic continuity rather than material remains. The Tibetan Plateau offers a different type of evidence. The Jahe mandible places a Denisovan-related individual at high elevation in a cold and oxygen-poor environment. This finding expands the ecological range associated with Denisovans. It suggests that some Denisovan populations were adapted to conditions beyond lowland forests and river valleys. The plateau environment imposes constraints on mobility and resource availability, implying seasonal movement or specialized strategies. Archaeological evidence in the region remains limited, but the fossil itself establishes presence rather than inference. Further east, no confirmed Denisovan fossils have been recovered from China beyond the Xiahe specimen. However, genetic studies of East Asian populations indicate Denisovan admixture events distinct from those affecting Oceanian populations. Some analyses propose at least two Denisovan lineages, separated by time or geography. This conclusion arises from differences in the Denisovan-derived DNA segments found in different modern populations. The interpretation does not require these lineages to have coexisted in the same place. It allows for separation across large distances and long periods. South Asia presents an intermediate picture. Genetic signals attributed to Denisovans are faint and inconsistent across populations. Some studies detect Denisovan-related ancestry in specific groups, while others do not. The variation may reflect multiple migration waves, later population mixing, or limitations in sampling. The archaeological record of South Asia during the relevant periods is complex, with long sequences of stone tool traditions that change gradually rather than abruptly. Without associated fossils or sediment DNA, Denisovan presence remains uncertain in this region. The absence of Denisovan fossils across much of Asia does not imply absence of Denisovan populations. Fossil preservation depends on geology, climate, and chance. Cold, stable environments like caves in Siberia favor DNA survival. Tropical and monsoon regions do not. As a result, the Denisovan fossil record is likely biased toward northern and high-altitude locations. Genetic signals in modern populations compensate for this bias to some extent, but they do so indirectly. Researchers often describe Denisovans as having a wide but sparse distribution. This phrase reflects the nature of the evidence. There are no known Denisovan settlement clusters or dense site concentrations. Instead, there are isolated points where genetic or fossil data intersect with archaeological contexts. Each point stands largely on its own. Connections between them are drawn through models rather than through continuous material trails. Environmental reconstructions support the possibility of such a distribution. During the Middle Pleistocene, large parts of Asia supported mixed environments capable of sustaining mobile hunter-gatherer populations. River systems provided corridors for movement, while mountain ranges created barriers that could separate groups over time. Denisovan-related populations, like other archaic humans, likely responded to these features through movement rather than through fixed territorial boundaries. The genetic legacy preserved in modern populations also suggests uneven survival. Denisovan ancestry persisted at higher levels in regions where later population replacement was limited. In areas with repeated waves of migration and mixing, Denisovan-derived segments were diluted or lost. This process does not reflect cultural dominance or technological superiority. It reflects demographic history, including population size, isolation, and continuity. Maps produced in academic publications often show shaded regions indicating inferred Denisovan range. These maps are explicitly labeled as approximations. They are derived from genetic gradients and from the location of a small number of fossils. The shading fades at the edges, acknowledging uncertainty. Such representations emphasize probability rather than certainty, and they change as new data are added. The concept of a Denisovan supercivilization does not appear in the technical literature. What appears instead is a pattern of wide dispersal without dense concentration. Denisovans are treated as populations adapted to varied environments, interacting with other human groups when routes overlapped. Their presence across Asia is reconstructed as a series of contacts and occupations, not as a unified expansion. The record remains open-ended. New sediment DNA studies may identify Denisovan presence at sites previously attributed only to other groups. New fossils may extend the known range further. Until then, the Denisovan footprint across Asia remains thin, defined by genetic persistence rather than by monuments, settlements, or named regions. Adaptation. Without architecture or records. Denisovan-related adaptations are identified through biological signals, rather than through built remains. No structures, roads, or tools clearly linked to environmental engineering have been attributed to Denisovans. Instead, adaptation is inferred from genetic variants that persist in modern populations and from the ecological contexts in which Denisovan fossils have been found. These forms of evidence are indirect and limited to what can be measured through comparison. One of the most closely examined examples involves high-altitude physiology. On the Tibetan Plateau, populations living at elevations above 4,000 meters show distinctive responses to low oxygen levels. Studies have focused on a region of the genome near EPAS1, a gene involved in hypoxia response. The variant common in Tibetan populations differs from most modern human versions but closely matches a Denisovan sequence. This correspondence is established through alignment and statistical testing, rather than through anatomical comparison. Research on this variant emphasizes its functional effect. Individuals carrying it tend to have lower hemoglobin concentrations than those without it. reducing the risk of chronic mountain sickness. The variant does not increase oxygen transport capacity. Instead, it moderates the body's response to hypoxia. Papers describing this relationship use controlled language, reporting association and mechanism without extending the finding beyond physiology. The adaptation is presented as a product of inheritance rather than as evidence of planning or design. High-altitude adaptation is not the only area where Denisovan-derived segments appear to persist. Other introgressed regions have been identified in genes related to immune response. Some Denisovan variants are found in toll-like receptor genes, which play a role in pathogen detection. These variants are unevenly distributed across populations and appear at low frequencies. Their functional significance is often uncertain. Where associations are proposed, they are described as modest effects rather than decisive advantages. The immune-related findings rely on large comparative datasets. Researchers examine patterns of variation across thousands of genomes, looking for segments that show Denisovan affinity and elevated frequency in certain populations. Statistical thresholds are applied to distinguish introgression from background variation. Even when a Denisovan origin is supported, the biological impact of the segment may remain unclear. Many introgressed regions appear neutral, neither strongly favoured nor selected against. Environmental breadth also informs discussions of Denisovan adaptation. Fossil and genetic evidence places Denisovan-related populations in cold northern regions, temperate zones, and high-altitude plateaus. This range implies flexibility rather than specialization tied to a single niche. Archaeological contexts associated with Denisovan DNA show use of caves and open sites similar to those used by other archaic humans. There is no indication of large-scale environmental modification. Dietary adaptation is difficult to reconstruct. Stable isotope analysis, which can reveal aspects of diet, has not been widely applied to Denisovan remains due to their scarcity. Faunal assemblages from Denisova cave include remains of large and small mammals typical of the region, suggesting hunting and scavenging patterns consistent with other Paleolithic groups. Without direct isotopic data from Denisovan bones, dietary inference remains indirect. Thermoregulation is another area considered in general terms. Occupation of cold regions implies behavioral and physiological responses to low temperatures. These could include clothing, fire use, and shelter selection, but such behaviors leave limited traces that can be specifically assigned to Denisovans. Genetic studies have explored variants related to skin pigmentation and fat metabolism, but clear Denisovan-specific patterns have not been established. Most findings emphasize overlap with Neanderthal and modern human variation. The concept of adaptation in this context remains narrowly defined. It refers to traits that improved survival or reproduction in particular environments, and that can be detected through genetic persistence. It does not refer to cumulative technological systems or to social organization. Denisovan-related adaptations are identified one variant at a time, each evaluated through its frequency, distribution, and potential functional role. Comparisons with Neanderthals highlight this distinction. Neanderthal adaptation is often discussed alongside skeletal traits, such as robust limb bones or nasal morphology. Denisovans, lacking a comparable fossil sample, are known primarily through molecular data. As a result, adaptation is framed as sequence variation rather than as body form. This difference reflects evidence availability rather than a fundamental difference in biology. The absence of written records or symbolic systems limits interpretation. No Denisovan inscriptions, figurative art, or standardized iconography have been identified. Ornaments found in Denisova cave are described without cultural attribution. They are treated as objects produced by someone present in the cave at a given time, not as expressions of a named group's identity. Adaptation in this sense remains a biological process rather than a cultural one. Researchers are careful to separate adaptation from advancement. A genetic variant that confers a local advantage does not imply a higher level of complexity. It reflects compatibility with specific conditions. Denisovan-derived adaptations are discussed alongside similar cases from Neanderthals and early modern humans, forming part of a broader pattern of gene flow and selection in human evolution. The persistence of Denisovan variants in modern populations depends on demographic factors. Small populations, geographic isolation, and limited later admixture can preserve introgressed segments longer. In regions with repeated population turnover, such segments may be lost. This process explains why Denisovan ancestry is concentrated in certain areas without requiring continuous occupation by Denisovan populations themselves. Laboratory techniques shape what can be detected. Advances in sequencing allow for finer resolution of introgressed segments. Functional studies using cell cultures or animal models can test the effects of specific variants, but such studies are limited in number. Most Denisovan-related adaptations remain identified through correlation rather than through experimental confirmation. The language used in scientific reports reflects this uncertainty. Adaptations are described as associated with or linked to Denisovan ancestry. Claims are framed within confidence intervals and effect sizes. Negative results, where Denisovan segments show no detectable function, are also reported. This balance maintains a descriptive approach focused on evidence rather than narrative. In this framework, Denisovans appear as contributors to the modern human gene pool in specific contexts. Their adaptations persist where they remain useful or neutral. They do not point to a lost system of knowledge or construction. They are biological residues, shaped by environment and time. The record remains open to revision. New genomes may reveal additional Denisovan-derived adaptations or reclassify existing ones. Fossil discoveries may provide anatomical context that reframes current interpretations. For now, adaptation without architecture defines the Denisovan presence, measurable, limited, and recorded in sequences rather than in structures, contact zones without clear boundaries. The interaction between Denisovans and other human groups is inferred through overlap rather than through defined borders. No site preserves a clear division where one population ends and another begins. Instead, contact is detected through shared genetic material, layered occupations, and the coexistence of different human signatures within the same regions over extended periods. These contact zones are reconstructed retrospectively, using evidence that does not record the moment of interaction itself. Denisova Cave provides one of the clearest examples of such overlap. Genetic analysis has shown that the cave was used at different times by Denisovans, Neanderthals, and modern humans. In some layers, DNA from more than one group appears within a relatively narrow span of sediment. This does not imply simultaneous occupation in a social sense. It indicates that the cave was reused across generations by different populations moving through the region. The cave functioned as a shared resource rather than as a territorial marker. One specimen from Denisova Cave illustrates direct biological contact. A bone fragment recovered from the site was identified as belonging to an individual whose mother was Neanderthal and whose father was Denisovan. The identification was made through genome-wide analysis, which showed roughly equal contributions from each group. The individual lived more than 90,000 years ago, based on the sediment context. This finding demonstrates that interbreeding was not limited to distant events, but occurred directly and repeatedly. The presence of such individuals complicates attempts to define populations as separate units. Genetic boundaries were permeable. Groups that appear distinct when viewed through average genetic differences were capable of producing offspring without apparent barriers. This biological compatibility does not imply shared culture or stable integration. It reflects contact at points where movement paths intersected. Beyond the Altai region, similar contact is inferred through modern human genomes. Denisovan-derived segments in present-day populations indicate that early modern humans encountered Denisovan-related groups as they expanded across Asia. The locations of these encounters are not directly known. Genetic models estimate timing based on segment length and recombination rates, suggesting multiple episodes of contact rather than a single event. Neanderthal contact with Denisovans is also evident in the Denisovan genome itself. Analyses show that Denisovans carry segments of Neanderthal ancestry, implying earlier interactions between these two archaic groups. The direction of gene flow varies across studies, but the consensus supports repeated contact. These interactions likely occurred in regions where the ranges of the two groups overlapped, such as parts of Central Asia. The archaeological record does not clearly separate these interactions. Stone tools associated with Neanderthals and Denisovans cannot be reliably distinguished on technological grounds alone. Similar reduction strategies and raw material choices appear across sites attributed to different populations. This overlap makes it difficult to associate contact with changes in tool form or technique. Archaeologists therefore avoid linking specific assemblages to interbreeding events. Environmental corridors play a role in understanding contact zones. River valleys, mountain passes, and ecotonal regions facilitate movement and increase the likelihood of encounter. The Altai Mountains, situated between Siberian plains and Central Asian steppe, represent one such corridor. Seasonal migration of animals and humans through these areas would have created repeated opportunities for contact without sustained cohabitation. Contact does not require long-term coexistence. A brief overlap in time and space is sufficient for genetic exchange. This principle is reflected in the patchy distribution of Denisovan ancestry in modern populations. Some groups show evidence of Denisovan admixture, while neighbouring populations do not. The pattern suggests localised contact, followed by divergence rather than widespread integration. In Southeast Asia, contact zones are inferred rather than observed. Genetic evidence indicates Denisovan admixture in populations whose ancestors passed through the region. Fossil evidence for Denisovans themselves is absent. Contact may have occurred in mainland regions now submerged or heavily altered, leaving no recoverable record. The lack of clear boundaries extends to chronology. Denisovans, Neanderthals, and modern humans overlapped in time for tens of thousands of years. Their interactions were not confined to a single transitional period. Instead, they occurred intermittently as populations shifted in response to climate, resources, and demographic pressure. This temporal overlap reduces the usefulness of linear replacement models. Genetic studies approach these interactions through statistical reconstruction. Models incorporate population sizes, divergence times, and migration rates. Contact is represented as gene flow parameters rather than as events with social context. The results are expressed as probabilities and ranges. They describe the presence of interaction without specifying its form. Some Denisovan-derived genetic segments in modern humans appear to have been selected against over time. These segments are underrepresented in functional regions of the genome, suggesting that not all introgression was neutral or beneficial. This pattern is consistent with limited compatibility, rather than complete integration. It supports the view that contact occurred without full population merging. The concept of a contact zone in this context is therefore abstract. It refers to regions where genetic exchange was possible, not to borders marked by material culture or physical barriers. These zones shifted over time as climates changed and populations moved. Archaeological sites that preserve long sequences provide the clearest windows into this process. Caves and rock shelters, used repeatedly over millennia, accumulate evidence of multiple populations without preserving their interactions directly. The stratigraphic record records succession, not dialogue. Genetics fills some of this gap by revealing biological exchange, but it does not reconstruct social relationships. Researchers emphasize that contact does not imply dominance or transmission of complex systems. Gene flow can occur through small-scale interactions, including encounters between individuals or small groups. The Denisovan contribution to modern human genomes is consistent with such interactions. It does not require large, mixed communities or sustained alliances. The absence of clear boundaries challenges older models of human evolution that relied on distinct stages and replacements. The Denisovan case supports a mosaic view where multiple human groups coexisted, interacted, and exchanged genes without losing their broader identities immediately. This view is built from cumulative evidence rather than from a single defining discovery. As methods improve, contact zones may be identified more precisely. Sediment DNA analysis may reveal overlapping presence at additional sites. More ancient genomes may clarify the direction and timing of gene flow. For now, the picture remains defined by overlap without lines. Denisovans appear within this picture as participants in a network of contact that spanned much of Eurasia. Their interactions with Neanderthals and modern humans are recorded in fragments of DNA rather than in shared material traditions. The contact zones remain without clear boundaries, shaped by movement and chance rather than by fixed divisions. Disappearance without a single moment. The Denisovan presence does not end at a clear boundary in the archaeological or genetic record. There is no layer that marks a final occupation, no horizon that separates a Denisovan past from a human present. Instead, the traces associated with Denisovans thin gradually. They become harder to detect, more fragmented, and increasingly indirect. Disappearance, in this case, is observed as a process rather than as an event. In Denisova Cave, the sediment sequence continues beyond layers that contain Denisovan DNA. Above them, material associated with later Upper Paleolithic occupations appears. These upper layers include stone tools and remains attributed to modern humans. The transition between layers does not indicate conflict or sudden abandonment. It reflects a change in who used the cave over time. Natural deposition continued. Humans came and went. The record does not preserve a moment of replacement. Genetic evidence mirrors this pattern. Denisovan-derived segments persist in modern human genomes, but only in specific regions and at varying frequencies. Over generations, recombination breaks these segments into smaller pieces. Some are lost entirely. Others remain because they are neutral, or because they offer a slight advantage in a particular environment. This gradual dilution creates a fading signal rather than an abrupt stop. The timing of Denisovan decline cannot be fixed to a single date. Estimates depend on models that incorporate mutation rates, recombination, and population size. These models suggest that Denisovan populations were already declining or fragmenting by the time modern humans expanded widely across Asia. The decline is inferred indirectly, from decreasing genetic contribution and from the absence of later Denisovan fossils, rather than from a dated terminal site. Environmental change provides one context for this process. During the late Pleistocene, climate oscillations altered habitats across Eurasia. Glacial advances and retreats reshaped vegetation zones, animal distributions, and migration routes. Populations adapted to specific ecological niches may have found those niches shrinking or shifting. For small dispersed groups, such changes could reduce population size without leaving a distinct archaeological signature. Demographic pressure from expanding modern human populations is another factor considered in broad terms. Modern humans entering Asia carried new technologies and social networks that supported larger population sizes. Increased numbers alone can influence survival outcomes, even without direct competition. In such scenarios, smaller populations may be absorbed through interbreeding or may decline through reduced access to resources. The Denisovan genetic legacy in modern humans is consistent with partial absorption rather than complete isolation. Absorption does not imply equality of representation. Genetic studies show that Denisovan ancestry makes up only a small fraction of modern genomes, even in populations where it is most concentrated. This suggests asymmetrical mixing, where Denisovan populations contributed genes but did not persist as distinct groups. Over time, their separate identity became biologically diluted. The archaeological record does not contradict this interpretation, but it does not confirm it directly either. Sites associated with Denisovan DNA do not show a clear replacement sequence. Instead, they show continuity of occupation with changing assemblages. Tools become more standardized in some regions. Ornamentation appears more frequently. These changes are attributed to broader trends in Upper Paleolithic archaeology, rather than to the disappearance of a specific group. In regions where Denisovan ancestry is highest today, such as Melanesia, the archaeological record shows continuity of modern human occupation over tens of thousands of years. There is no evidence for a separate Denisovan material culture persisting alongside modern humans into the late prehistoric period. The Denisovan contribution appears to have been incorporated early and then carried forward biologically rather than culturally. The absence of late Denisovan fossils is a key limitation. Without skeletal remains from later periods, it is not possible to track changes in Denisovan anatomy or health over time. This absence may reflect true disappearance, but it may also reflect preservation bias. Regions where Denisovans likely lived, based on genetic inference, include tropical and subtropical areas where fossil preservation is poor. The record may be incomplete rather than conclusively empty. Researchers approach this uncertainty by framing disappearance as a fading signal. Archaeological attribution becomes impossible without biological markers. Each line of evidence weakens at a different rate. Together, They suggest a gradual loss of distinct Denisovan populations rather than a catastrophic end. Comparisons with Neanderthals are often made cautiously. Neanderthals have a richer fossil record and a clearer terminal period in parts of Europe. Denisovans lack this resolution. Their disappearance cannot be mapped onto a single region or date. It must be inferred across multiple regions, each with its own environmental and demographic history. Some genetic segments of Denisovan origin appear to have been selected against in modern human populations. These segments are underrepresented in genes associated with male fertility and other essential functions. This pattern suggests that not all Denisovan modern human combinations were equally viable over the long term. Selection against certain introgress segments would contribute to the gradual erosion of Denisovan genetic presence without requiring a population collapse. Other segments persisted because they had no negative effect or because they conferred a local advantage. This uneven survival creates a patchwork legacy. Denisovans did not vanish completely. Their genetic material remains, scattered and reduced, integrated into genomes that no longer carry a Denisovan identity. The term extinction is therefore used carefully in this context. It refers to the loss of Denisovans as a distinct population, not to the elimination of all Denisovan ancestry. This distinction is emphasized in technical writing. Denisovans ceased to exist as a separate group, but their biological traces continued through admixture. Archaeological silence follows this biological fading. Without distinctive tools, settlements, or art tied uniquely to Denisovans, their disappearance leaves no clear material boundary. Later sites cannot be assigned to Denisovans or excluded from them with confidence. The identity dissolves into uncertainty. This mode of disappearance challenges expectations shaped by later historical periods, where societies leave records of collapse or conquest. In deep prehistory, populations can vanish without documentation. Their absence is detected only through comparison, through noticing what no longer appears where it once did. The Denisovan case illustrates this pattern clearly. Early presence is inferred through rare fossils and genetic markers. Interaction is detected through admixture. Decline is inferred through dilution and absence. No single site records the end. No artifact marks a final moment. The record remains open to revision. A new fossil could extend the Denisovan timeline. A new genome could reveal later survival in an unexpected region. Until then, disappearance is understood as a long process shaped by environment, demography, and chance. What remains is a thin residue, measurable but incomplete. Denisovans fade from view not through a sudden break, but through gradual integration and loss. Their disappearance has no single moment because it did not occur all at once. Fragments that survived in living populations. Denisovans are no longer identified through living communities or distinct physical remains. These fragments are unevenly distributed, reduced in size, and shaped by long processes of recombination and population change. What survives does so quietly, embedded within genomes that no longer carry a Denisovan identity. The detection of Denisovan ancestry in living populations depends on comparison. African genomes are used as a baseline because they are assumed to contain little or no Denisovan ancestry. Deviations from this baseline, when matched closely to Denisovan sequences, indicate ancient admixture. The method identifies similarity, not descent in a social or cultural sense. The highest concentrations of Denisovan ancestry are found in Melanesian populations, particularly in Papua New Guinea and surrounding islands. In some groups, Denisovan-derived DNA makes up several percent of the genome. This proportion is small in absolute terms, but large in comparison to other regions. The persistence of these fragments reflects demographic continuity. Populations in these areas experienced fewer later migration events that would dilute earlier genetic contributions. In East Asia, Denisovan ancestry appears at much lower levels. The fragments detected are shorter and less frequent. This pattern is consistent with older admixture events, followed by extensive recombination and later population mixing. The genetic signal is present, but it is diffuse. Large sample sizes are required to detect it reliably, and results vary depending on the reference datasets used. South Asia presents a mixed picture. Some populations show faint Denisovan-related signals, while others show none. The variation reflects complex population history, including multiple migration waves and regional isolation. Genetic studies in this region emphasize uncertainty, noting that Denisovan ancestry may be underestimated due to limitations in sampling and reference genomes. The survival of Denisovan fragments is shaped by recombination. Each generation reshuffles genetic material, breaking inherited segments into smaller pieces. Over tens of thousands of years, this process reduces long Denisovan segments into short fragments. Many are lost entirely through chance. Others persist because they lie in regions of the genome that are neutral or only weakly affected by selection. Natural selection further filters these fragments. Denisovan-derived segments that reduce reproductive success or interfere with essential biological processes tend to be removed over time. Studies show that Denisovan ancestry is underrepresented in certain functional regions of the genome, such as those involved in male fertility. This pattern suggests that some Denisovan modern human combinations were disadvantageous in the long term. Other fragments persist because they have little effect. Neutral variants can remain in a population simply because they do not impose a cost. Their survival is governed by population size and structure rather than by adaptive value. In small or isolated populations, neutral Denisovan fragments are more likely to persist than in large, rapidly mixing populations. A smaller subset of Denisovan-derived variants appears to have been favored under specific environmental conditions. High-altitude adaptation in Tibetan populations remains the most frequently cited example. In this case, a Denisovan-related variant rose in frequency because it improved survival in a particular setting. Such cases are rare. Most Denisovan fragments do not show clear signs of positive selection. The presence of Denisovan DNA in modern genomes does not imply continuity of Denisovan populations. Once Denisovan groups interbred with modern humans, their descendants became part of modern human populations. Over time, Denisovan ancestry was diluted as those populations mixed with others. The biological identity dissolved even as some genetic material persisted. Genetic fragments do not preserve information about language, social structure, or behavior. They record inheritance, not culture. A Denisovan-derived gene segment does not indicate that its carrier follows Denisovan traditions or practices. It simply marks a historical interaction that occurred far in the past. The uneven distribution of Denisovan ancestry highlights the role of demographic history. Regions that experienced repeated waves of migration tend to show lower levels of Denisovan DNA. Regions with longer periods of relative isolation show higher levels. This pattern does not require Denisovans to have been more prevalent in isolated regions. It reflects what happened after contact, not where contact occurred. Analytical methods influence what is detected. Different studies use different Denisovan reference genomes, including sequences from the Denisova cave specimens and reconstructed Denisovan haplotypes. Variations in methodology can change estimates of Denisovan ancestry by small but meaningful amounts. Researchers report these differences and treat ancestry proportions as approximations rather than precise values. The length of Denisovan-derived segments provides information about timing. Shorter segments generally indicate older admixture events. In most populations, Denisovan segments are short, supporting the view that admixture occurred tens of thousands of years ago. Longer segments are rare and do not indicate recent Denisovan ancestry. They reflect statistical variation rather than later survival of Denisovan populations. Genetic studies also show that Denisovan ancestry is not evenly distributed across individuals within a population. Some individuals carry more Denisovan fragments than others. This variation arises through inheritance patterns and does not correlate with visible traits. It is detectable only through sequencing and analysis. The persistence of Denisovan fragments complicates simple narratives of replacement. Modern humans did not fully replace Denisovans without interaction. They absorbed part of their genetic material. The result is a layered genome that reflects multiple past populations. Denisovans are one of several archaic groups whose traces remain in this way. These findings have reshaped how human ancestry is described. Instead of discrete stages, the record shows overlap and mixture. Denisovan fragments in living populations serve as evidence of this process. They demonstrate that ancient human diversity did not disappear entirely, but was redistributed through admixture. The language used in scientific descriptions remains restrained. Denisovan ancestry is described in percentages, confidence intervals, and segment lengths. Researchers avoid assigning significance beyond what the data support. Modern populations carrying Denisovan ancestry do not form a separate category. They are part of the broader variation within Homo sapiens. Denisovan fragments contribute to this variation without defining it. Their presence is one element among many shaping genetic diversity today. As sequencing technologies improve, additional Denisovan fragments may be identified. New reference genomes could refine estimates reveals. Some fragments currently classified as Denisovan may be reassigned as belonging to related but distinct archaic groups. The picture remains provisional. What persists is a pattern of survival through inheritance. Denisovans did not leave cities, monuments, or written records. They left fragments of DNA that continue to replicate with each generation. These fragments are silent. They do not explain themselves. They remain detectable only through comparison and statistical analysis. The Denisovan presence in living populations is therefore partial and indirect. It does not reconstruct a society. It records contact and absorption. The fragments that survive did so by chance, neutrality, or limited advantage. Together, they form a quiet residue of a population that no longer exists as a separate group. Limits of what can be recovered. Research on Denisovans is defined as much by absence as by presence. A small number of fossils, fragments of DNA, and a limited set of archaeological contexts form the basis of current knowledge. Each method used to recover information carries constraints that shape what can be said and what must remain unresolved. The fossil record is the most visible limitation. Only a few Denisovan specimens have been identified with confidence. These include a finger bone and several teeth from Denisova cave, along with a mandible from the Tibetan plateau. The small number of remains restricts anatomical reconstruction. There is no complete Denisovan skeleton. Body proportions, facial structure, and variation within the population cannot be described directly. Any attempt to visualize Denisovans relies on extrapolation from genetic similarity to Neanderthals and modern humans rather than on physical evidence. Preservation bias affects where fossils can survive. Cold, stable environments favor the survival of bone and DNA. Much of Asia, where Denisovan-related populations are inferred to have lived, includes regions with acidic soils, high humidity, or intense biological activity. These conditions degrade organic material rapidly. As a result, Denisovan fossils may have existed in many regions without leaving recoverable traces. The absence of fossils therefore reflects environmental loss as much as historical reality. Genetic evidence, while powerful, also has limits. Denisovan genomes are reconstructed from fragmentary DNA, preserved under specific conditions. The referenced Denisovan genome is based on a small number of individuals. It cannot capture the full genetic diversity that likely existed among Denisovan populations, spread across a vast area. When modern genomes are compared to this reference, only similarity to the sampled Denisovan individuals can be detected. Denisovan-related populations that differ genetically from those individuals may leave signals that are misclassified or missed entirely. Modern population sampling introduces additional bias. Genetic studies rely on available samples, which are unevenly distributed across the world. Some regions are well represented, while others are underrepresented or absent. This imbalance affects estimates of Denisovan ancestry. Regions with sparse sampling may appear to lack Denisovan signals, when, in fact, those signals have not been adequately searched for. Researchers acknowledge this limitation and treat geographic patterns as provisional. Temporal resolution is another constraint. Genetic methods estimate timing indirectly through mutation rates and recombination patterns. These estimates are expressed as ranges rather than precise dates. Small changes in assumed mutation rates can shift estimated admixture events by thousands of years. As a result, timelines remain approximate. Archaeological dating methods also carry uncertainty, especially in older contexts where radiocarbon dating is no longer effective. Sediment DNA analysis has expanded the range of detectable presence, but introduces its own challenges. DNA fragments recovered from soil can confirm that a population was present in a location, but they do not indicate how long the population stayed or what activities took place. Sediment can be disturbed by natural processes, mixing material from different periods. Careful controls reduce error, but interpretation remains cautious. Presence does not equal occupation intensity or cultural association. Attribution of artifacts presents a further limitation. Stone tools and ornaments cannot be assigned to Denisovans with certainty unless directly associated with Denisovan DNA or fossils. Many tool types found in Denisovan-related layers are also found elsewhere in contexts attributed to Neanderthals or modern humans. This overlap prevents the definition of a distinct Denisovan material culture. Archaeological descriptions therefore focus on form and technique, rather than on authorship. The absence of written records or symbolic systems removes another avenue of inquiry. Without inscriptions, art styles, or standardized symbols clearly linked to Denisovans, cultural reconstruction is limited. Objects found in Denisova cave are described without assuming shared meaning or tradition. This restraint avoids speculation, but leaves questions about social organization, communication, and belief systems unanswered. Comparative frameworks also impose limits. Denisovans are often discussed in relation to Neanderthals and modern humans because these groups are better documented. This comparison shapes interpretation. Traits or patterns that differ from those groups may be overlooked or forced into existing categories. The Denisovan record may represent variation that does not fit neatly into known models of human behavior. The concept of Denisovans itself reflects methodological boundaries. The name refers to a genetic cluster identified through analysis, not to a population defined by self-recognition or shared identity. Denisovans are defined by difference from other genomes, not by internal cohesion. This approach is useful for classification, but limits insight into how Denisovan-related populations may have understood themselves or related to others. Uncertainty also arises from the possibility of additional archaic populations. Genetic analyses suggest that Denisovans themselves may have interacted with an older, unidentified hominin group. This inference is based on deep genetic divergence within the Denisovan genome. Without fossils or independent confirmation, the proposed population remains hypothetical. Its existence complicates interpretation by adding another layer of unknown variation. Technological limits shape outcomes as well. Sequencing ancient DNA requires well-preserved material and extensive contamination control. Not all fossils can be sequenced successfully. Failed attempts are rarely emphasized in public summaries but are common in practice. The resulting dataset reflects what can be extracted, not what once existed. Advances in technology may recover information from samples that are currently unusable, but such advances cannot be assumed. Interpretive restraint is therefore a defining feature of Denisovan research. Scientific papers emphasize confidence intervals, error margins, and alternative explanations. Claims are limited to what can be supported by data. Where evidence is lacking, uncertainty is stated rather than filled. This approach contrasts with broader narratives that extend beyond the available record. The limits of recovery also affect how disappearance is understood. Without late Denisovan fossils or clear terminal sites, the end of Denisovan populations cannot be described in detail. Genetic dilution suggests absorption, but the process cannot be observed directly. Archaeology does not provide a sequence of decline. The disappearance remains inferred rather than documented. These constraints do not invalidate the Denisovan record. They define its scope. The available evidence establishes Denisovans as a real and distinct part of human evolutionary history. It also sets clear boundaries on interpretation. Denisovans can be located genetically and, in a few cases, geographically. Beyond that, much remains unknown. Future discoveries may alter this balance. New fossils, improved sediment analysis, and expanded genetic sampling could fill some gaps. At the same time, certain questions may remain permanently unresolved due to the nature of deep prehistory. The record is shaped by what survives, not by what once existed in full. Denisovan research, therefore, proceeds within limits that are acknowledged and measured. Each new finding adds detail without transforming the overall picture into a complete narrative. The evidence accumulates slowly, constrained by preservation, technology, and method. What can be recovered is partial. What cannot be recovered defines the edges of understanding. The Denisovan story is written within these boundaries, not as a finished account, but as an outline formed by fragments, comparisons, and careful restraint. When evidence is replaced by interpretation, outside academic research, Denisovans are often presented in forms that differ from the published record. The shift does not occur through new data, but through changes in emphasis and framing. Limited evidence is expanded into coherent stories, and provisional models are treated as settled conclusions. This process does not require fabrication. It relies on selection, omission, and simplification. Scientific descriptions of Denisovans are cautious by design. They describe genetic relationships, statistical confidence, and uncertainty. In public-facing summaries, these elements are often compressed. Technical language is replaced with accessible terms, and conditional statements are shortened. Over time, repetition of simplified descriptions can produce the impression of completeness, even when the underlying evidence remains sparse. One common transformation involves scale. Genetic signals that represent a few % of ancestry are sometimes described as dominant contributions. The term Denisovan shifts from a genetic reference to a population label and then to a cultural identity. This shift occurs gradually. It does not depend on a single claim, but on accumulated phrasing that treats Denisovans as a unified group with shared characteristics beyond what can be supported. Maps offer another example. Academic maps showing inferred Denisovan range are usually shaded, labelled as approximate, and accompanied by notes on uncertainty. In secondary accounts, these maps are sometimes redrawn with sharper boundaries and stronger colours. The visual language implies territory rather than probability. The viewer may infer settlement or control where the original data indicate only genetic similarity. Material artifacts also acquire expanded meanings. Objects from Denisova cave, such as the polished stone bracelet, are sometimes described as evidence of advanced craftsmanship or symbolic culture. In technical reports, these objects are discussed in terms of material, manufacturing traces, and stratigraphic context. When removed from this framework, the same objects are presented as indicators of complex social systems. The change lies in interpretation, not in the artifact itself. Chronology is another area where interpretation replaces evidence. Genetic admixture dates are expressed as ranges, often spanning several thousand years. In popular accounts, these ranges are narrowed or replaced by specific dates. The result is a timeline that appears precise, but is not supported by the data. This precision can suggest coordination or planning where none has been demonstrated. Language plays a central role in this transformation. Terms like superior, advanced, or lost civilization do not appear in scientific literature on Denisovans. When these terms are introduced, they carry assumptions about hierarchy and development. They imply comparison to later societies and technologies that did not exist in the Paleolithic context. The evidence does not require such comparisons, but they persist because they provide narrative clarity. The absence of material evidence can also be reinterpreted. In research, lack of artifacts or settlements is treated as a limitation of preservation and sampling. In interpretive accounts, the same absence is sometimes framed as deliberate concealment or destruction. This shift assigns intention to processes that are better explained by environmental decay and demographic change. It replaces geological and archaeological explanation with narrative inference. Genetic adaptation is similarly reframed. Denisovan-derived variants associated with specific environments are sometimes described as proof of specialized knowledge or engineering. In the scientific record, these variants are discussed as inherited traits shaped by selection. They do not imply understanding of genetics or physiology. The interpretive leap occurs when biological outcomes are treated as products of conscious design rather than of evolutionary process. Visual reconstructions contribute to this effect. Illustrations of Denisovans often depict uniform appearance, clothing, and setting. These images are produced to communicate ideas quickly, but they can imply standardization that is not supported by evidence. With no complete Denisovan skeleton and limited anatomical data, such reconstructions are speculative. They become influential through repetition rather than through validation. Another layer of interpretation arises from comparison with later civilizations. When Denisovans are described using terms drawn from agricultural or urban societies, such as engineers or builders, the comparison introduces anachronism. Paleolithic populations operated within different constraints and opportunities. Applying later categories obscures these differences and reshapes the evidence to fit familiar frameworks. The process is cumulative. Individual simplifications may seem minor, but together they alter the overall picture. What begins as a genetic cluster becomes a people, then a culture, then a civilization. Each step moves further from the evidence while retaining its authority. The original uncertainties are rarely restated, and the provisional nature of models is lost. Academic responses to this process are generally restrained. Researchers continue to publish within methodological limits and often include explicit statements about what their findings do not show. These statements are less frequently quoted than headline results. As a result, the corrective context remains within specialist literature, while interpretive narratives circulate more widely. This gap between evidence and interpretation is not unique to Denisovan research. It appears in many areas of deep prehistory, where data are fragmentary and methods are indirect. The Denisovan case is distinctive only in the extent to which genetic discovery preceded archaeological context. This sequence created a population category before its material record could be described, leaving space for interpretation to expand. The replacement of evidence with interpretation does not require deliberate misrepresentation. It arises from the need to make complex findings accessible and meaningful. However, accessibility can come at the cost of accuracy when uncertainty is removed. Within the scientific record, Denisovans remain defined by limits. They are identified through genetic difference, a few fossils, and scattered archaeological associations. Outside that record, they are often described through coherence that has not been demonstrated. This distinction matters because interpretation shapes expectation. When Denisovans are presented as a lost civilization, their absence of cities, records, and technologies requires explanation. The scientific record does not pose this problem because it does not claim those features existed. The problem emerges only when interpretation exceeds evidence. Researchers emphasize that deep prehistory rarely produces complete pictures. Populations appear and disappear without leaving comprehensive traces. Denisovans fit this pattern. Their study is constrained by what survives, not by what later observers wish to find. Interpretation that moves beyond this constraint risks creating narratives that cannot be tested. The academic approach treats evidence as incomplete by default. Models are revised as new data appear. Interpretive accounts tend to solidify early impressions into fixed claims. This divergence explains why Denisovans can appear differently depending on the source, even when all accounts reference the same discoveries. The replacement of evidence with interpretation is therefore not a separate phenomenon. It is a transformation that occurs as information moves away from its original context. In the case of Denisovans, the transformation is especially visible because the evidence is minimal and the implications are broad. What remains consistent in the scientific record is restraint. Denisovans are presented as part of a complex evolutionary landscape, interacting with other human groups and leaving limited traces. Where evidence ends, description stops. Interpretation begins only when that boundary is crossed. The Denisovan case illustrates how easily that boundary can be blurred. It shows how absence invites narrative, and how provisional findings can be treated as final. The challenge lies not in rejecting interpretation, but in distinguishing it from evidence. What the record allows and what it does not. The Denisovan record permits certain statements and resists others. This distinction does not depend on preference or interpretation. It follows from the kinds of data that exist and the methods used to study them. Where evidence is direct, claims can be made with caution. Where evidence is indirect or absent, statements remain limited or excluded entirely. The record allows Denisovans to be identified as a distinct archaic human population. This identification rests on genomic data recovered from fossil remains and sediment samples. The genetic differences between Denisovans, Neanderthals, and modern humans are measurable and repeatable. These differences are sufficient to define Denisovans as a separate group within the broader human family. This claim is supported by multiple independent studies using different analytical approaches. The record also allows Denisovans to be placed in time. Genetic divergence estimates indicate that Denisovans separated from the lineage, leading to Neanderthals and modern humans hundreds of thousands of years ago. Fossil and sediment contexts show Denisovan presence extending into the late Pleistocene. These temporal placements are expressed as ranges rather than exact dates. They reflect uncertainty in mutation rates and dating methods, but they establish broad chronological boundaries. Geographical placement is allowed in limited form. Denisovans are directly attested in southern Siberia and on the northeastern edge of the Tibetan Plateau. Beyond these locations, their presence is inferred through genetic signals in modern populations. This inference supports a wide distribution across parts of Asia. The record allows this conclusion because genetic admixture requires physical contact. It does not allow precise mapping of Denisovan settlements or migration routes. The record allows interbreeding between Denisovans and other human groups to be stated as fact. Genomic data show Denisovan ancestry in modern human populations and Neanderthal ancestry within Denisovan genomes. A first-generation Denisovan Neanderthal individual provides direct confirmation. These findings establish biological interaction. They do not, however, describe the social conditions under which such interactions occurred. Physiological contributions are also supported in specific cases. Certain genetic variants in modern populations can be traced to Denisovan ancestry and linked to measurable effects, such as high-altitude adaptation. These associations are supported by genetic alignment and functional studies. The record allows these traits to be described as inherited advantages under particular environmental conditions. It does not allow broader claims about Denisovan knowledge or intentional design. The archaeological record allows limited association between Denisovans and material culture. Objects and tools found in Denisova cave appear in layers that contain Denisovan DNA. This association indicates that Denisovans were present when those objects entered the deposit. It does not allow exclusive attribution of those objects to Denisovans, nor does it define a distinct Denisovan cultural tradition. The record permits description of artifacts, not assignment of authorship beyond context. What the record does not allow is reconstruction of Denisovan society. There is no evidence for social organization, kinship systems, leadership structures, or group identity as understood in later human societies. Genetic data do not encode social roles. Archaeological materials are too sparse and mixed to support such reconstructions. Any claim about Denisovan social complexity lies outside recoverable data. The record does not allow claims of Denisovan civilization in the sense used for agricultural or urban societies. There is no evidence of permanent settlements, large-scale construction, agriculture, writing, or administrative systems. The environments in which Denisovans lived and the technologies associated with the period do not support such features. The absence is not explained by loss or destruction. It reflects the Paleolithic context itself. Technological advancement beyond known Paleolithic capacities is also not supported. Tools associated with Denisovan contexts fall within the range observed for other archaic and early modern human groups. They show skill and variation, but not technologies that exceed the period's constraints. The record allows recognition of competence. It does not allow attribution of anomalous or out-of-context technology. The record does not allow physical reconstructions with high confidence. With no complete skeleton and only a few isolated bones and teeth, Denisovan appearance cannot be described in detail. Estimates of stature, facial structure, or body proportions remain speculative. Visual reconstructions used in educational or media contexts are interpretive aids rather than evidence-based representations. Language and symbolic communication also fall outside what can be recovered. No inscriptions, symbolic systems, or repeated iconography have been identified. Ornaments found in Denisova cave cannot be linked to shared meaning or language. The record does not allow determination of why Denisovans disappeared as a distinct population. Genetic dilution, environmental change, and demographic pressure are consistent with the evidence, but none can be confirmed as a sole cause. The disappearance is inferred from absence and genetic absorption, rather than observed through terminal events. The record permits explanation in general terms, not definitive conclusions. Intentional concealment or suppression of Denisovan evidence is not supported. Gaps in the record are explained by preservation bias, sampling limits, and the deep timescales involved. Archaeological loss is common in Paleolithic contexts. The record allows explanation through natural processes. It does not allow claims of deliberate erasure. The boundary between what is allowed and what is not is maintained through method. Genetic studies rely on statistical thresholds and reproducibility. Archaeological studies rely on stratigraphy, dating, and material analysis. Claims that exceed these methods cannot be tested. As a result, they remain outside the scientific record. This boundary is not static. As new methods develop, what can be recovered may expand. Sediment DNA analysis has already extended detection beyond visible fossils. Improved sequencing may reveal additional Denisovan diversity. These advances may allow new statements to be made in the future. They do not retroactively support claims made without evidence. The Denisovan record therefore functions as a constrained framework. It supports certain core facts: existence, distinction, interaction, and partial genetic legacy. It resists expansion into areas where data are insufficient. This resistance is not a flaw. It is a property of the evidence. Misunderstanding arises when the absence of evidence is treated as an invitation rather than a limit. In the scientific approach, absence marks uncertainty. In interpretive narratives, absence is often filled. The record itself does not do this. It remains silent where it cannot speak. The discipline of restraint is central to Denisovan research. Researchers regularly state what their findings do not show. These statements are as important as the findings themselves. They prevent the record from being used to support claims it cannot bear. Understanding Denisovans requires accepting incompleteness. The available data form a partial outline, not a finished picture. This outline is reliable, where it is defined, and undefined where it is not. The distinction matters because it preserves the integrity of the past as something discovered rather than invented. What the record allows is limited but solid. What it does not allow is extensive but tempting. The difference between the two defines the Denisovan case. It shows how deep history is reconstructed through fragments and how easily those fragments can be overextended. Within these limits, Denisovans remain an established part of human evolutionary history. They are known through genetics, a few fossils, and cautious archaeological association. Beyond that, the record ends. Where it ends, interpretation begins, but evidence does not follow. The Denisovan record stands as an example of how far evidence can go and where it must stop. It does not close questions. It sets boundaries around them. Those boundaries are what give the existing knowledge its strength, a population known only by traces. Denisovans are identified without a continuous record. They are known through fragments that do not assemble into a complete picture. The evidence that defines them is indirect, distributed across different fields, and separated by gaps that cannot be filled with certainty. What remains is a population outlined by traces rather than by a preserved narrative. The primary traces are genetic. Denisovans are first recognized through differences in DNA extracted from a small number of ancient remains. These differences are defined statistically by comparing sequences and measuring divergence. The result is a genetic category that exists independently of cultural description. Denisovans are known as a population because their genomes cluster together and differ from those of Neanderthals and modern humans. This definition does not require knowledge of how they lived, only that they existed as a biologically distinct group. Fossil traces provide limited physical context. The Denisovan remains recovered so far include isolated bones and teeth. These specimens do not form a representative sample. They do not allow assessment of variation within the population. Age, sex, health, and physical appearance cannot be reconstructed beyond narrow observations tied to individual specimens. Each fossil adds a point of reference, but not a framework. The archaeological traces associated with Denisovans are contextual rather than diagnostic. Tools and objects appear in layers that contain Denisovan DNA, but those same layers may also contain evidence of other human groups. The artifacts themselves do not carry identifying marks. They cannot be assigned to Denisovans without additional biological association. As a result, material culture remains descriptive, not attributive. Environmental traces shape interpretation indirectly. Denisovan-related evidence appears in regions with diverse ecological conditions, including cold mountain zones, temperate forests, and high-altitude plateaus. These settings indicate that Denisovans were capable of occupying varied environments. They do not specify how Denisovans organized their movements or resources. The landscape provides constraints, not explanations. Temporal traces are expressed as ranges rather than sequences. Denisovan presence spans long periods, but the density of evidence varies. There is no continuous timeline that tracks Denisovan populations from emergence to disappearance. Instead, there are points in time where Denisovan DNA or fossils appear, separated by intervals where no direct evidence exists. Continuity is inferred, not observed. The Denisovan name itself reflects this condition. It is derived from a cave, not from a historical designation. The name does not encode identity, as understood by the population it labels. It is a reference created for classification. Denisovans are named after where they were discovered, not after how they referred to themselves or how they related to others. Genetic traces in living populations extend Denisovan presence beyond their physical disappearance. These traces persist unevenly. They are broken into small segments and distributed across individuals and populations. They do not reconstruct a Denisovan genome in full. They preserve only fragments that survived recombination, selection, and demographic change. Each fragment points backward, but none carries context on its own. The persistence of these fragments does not imply continuity of Denisovan identity. Genetic inheritance does not preserve social structure, belief, or memory. A Denisovan-derived variant in a modern genome does not transmit Denisovan experience. It transmits sequence information only. The trace is biological, not cultural. Sediment DNA adds another category of trace. It detects presence without bodies. This method expands detection but narrows interpretation. Sediment DNA cannot distinguish duration, frequency, or purpose of presence. It records that someone was there, not what they did. Together, these traces create a record defined by limits. Each method captures a different aspect of Denisovan existence, but none captures the whole. Genetics defines relatedness. Fossils define physical reality in minimal form. Archaeology defines activity without identity. Environment defines constraint without intention. Time defines range without sequence. The absence of integrated traces prevents synthesis into a narrative. There is no evidence for shared symbols, repeated architectural forms, or standardized practices across regions. The record does not allow identification of a Denisovan way of life beyond what is common to Paleolithic populations more broadly. This condition is not unique. Many ancient populations are known only through partial traces. Denisovans differ mainly in how their discovery occurred. Genetic identification preceded archaeological context. As a result, the population is defined biologically before it is defined materially. This inversion shapes how Denisovans are understood and discussed. The reliance on traces affects certainty. Each new finding modifies probabilities rather than confirming conclusions. Denisovan research advances through refinement rather than resolution. Models are updated as new genomes are added. Geographic inferences shift as sampling improves. The outline changes at the edges while remaining incomplete at the center. This incompleteness influences how Denisovans are positioned within human history. They are recognized as contributors to genetic diversity rather than as actors within documented events. Their role is measured through influence on later populations, not through preserved actions of their own. The absence of Denisovan records does not imply insignificance. It reflects the conditions under which evidence survives. Paleolithic populations operated in contexts that rarely produced durable traces. Most human lives from this period left no recoverable record. Denisovans are unusual, only in that some of their traces persisted long enough to be detected. Scientific caution maintains the distinction between trace and inference. Researchers describe Denisovans in terms of what can be detected. They avoid filling gaps with assumptions. This restraint preserves the reliability of the record, even as it limits explanatory depth. The Denisovan case illustrates how modern methods can identify populations that would otherwise remain unknown. It also shows the limits of those methods. Genetics can reveal difference and connection, but not experience. Archaeology can reveal activity, but not identity. Fossils can reveal presence, but not narrative. What remains is a population known through its effects rather than through its own record. Denisovans are visible where they intersect with others, where their DNA enters modern genomes, and where their remains survive under specific conditions. They are invisible where preservation failed or where contact did not occur. This mode of knowledge reshapes how deep history is approached. It prioritizes comparison over continuity. It accepts partial reconstruction as the norm rather than as a failure. Denisovans are not an exception to this pattern. They are a clear example of it. The study of Denisovans continues within these boundaries. New traces may appear. Others may be reinterpreted. The population will remain defined by fragments rather than by completeness. This condition is stable, not temporary. Denisovans do not emerge from the record as a fully described people. They remain an outline formed by what survives. Their existence is established. Their interactions are measurable. Their disappearance is inferred. Beyond that, the record does not extend. A population known only by traces is not silent because it lacked complexity. It is silent because time removes most evidence. Denisovans are present in the record where chance and environment allowed preservation. Elsewhere, they are absent without explanation. The Denisovan story, as it stands, is not a narrative of rise or fall. It is a description of detection. It shows how modern science encounters the past through fragments, and how those fragments define the limits of knowledge. Within those limits, Denisovans occupy a fixed place in human evolutionary history. They are neither hypothetical nor fully known. They exist as a population identified through traces, constrained by evidence, and understood through comparison. This condition does not invite closure. It reflects the nature of deep time. The record remains incomplete, and Denisovans remain known only by what persists. If you'd like more documentaries like this, you can subscribe to the channel. It helps support the work and makes sure you don't miss future uploads. You can also leave a like if the video was helpful. If you have a subject you want explored next, feel free to leave it in the comments. Thanks for watching.